Hello, everyone. It's my pleasure to welcome you all to this hands-on and demos session of our online course. So this is going to be the last lecture of this course. And in this lecture, like I had mentioned in our previous lectures, we'll be having some quick tasks and some demo sessions on how are we going to perform those tasks. So before moving on with the tasks directly, so I would like to take you through or give you a very brief overview on all the reference repositories that is publicly available databases, which gives very valuable information on the blood group related genetic variants. So when I say that, the very first mention goes to the ISBT resource, which is the International Society of Blood Transfusion. This has been the pioneering organization which systematically maintains, collects the information about newly reported and discovered blood group variants, and it also provides extensive information about these blood group variants, which is deposited publicly for the use of for people's use also. So this is how the front page looks like. It has multiple tabs and options to surf through, like the events, membership, communities, working parties, and resources. So we need to go into this working party section where we will be surfing through the red cell immunogenetics and blood group terminology working party. So this is where all the information related to blood group related variants will be found. So when we enter into this particular page, it provides a very quick introduction where we tend to see these three links: the blood group systems, antigens in systems, and blood group allele tables. So by clicking into each of these, we will be taken to a new page which provides all the information regarding that particular title. So first, when we talk about the blood group systems, we will be taken to something like this, which gives a tabulation in the form of a PDF. It gives information about all the blood group system names, what is the symbol, which gene is associated with each of these blood groups, what is the LRG ID of that particular gene, how many antigens are there in that particular blood group system. Again, coming back to the gene which is responsible, what is the chromosomal location of that particular gene, the CD number, etc. So this information is provided in the form of a PDF, and it can be, it is an option to download is also provided. And also when we look at antigens in system and blood group allele tables, we will be taken to two different pages which gives information in this format. So if we look at the antigens in the system, again, it provides a tabulation in the form of a PDF. It gives the list of blood group system symbols, how many antigens are present, what is the name of each of the antigen. For example, if we take the ABO blood group system, total number of antigens in that particular system is 4 antigens. What are they? A, B, both AB and A1. Similarly, for each of the 44 blood group systems, the total number of antigens and what is each of those antigens, the numbering of each of those antigens are also listed in the form of a table. So if at all there's a new discovery of, if at all there's a new antigen which is reported, if it has been placed under a new blood group system, this page will be constantly being updated by the people who are working for that particular party. And similarly, talking about the blood group allele tables, when we enter into that page, it gives a complete list of tabs like this. All the 44 blood group systems will be provided as tabs. And by pressing into or by clicking into each of these tabs, we get to access the blood group related variants in each of these blood group systems. For example, if we take the ABO blood group system, it provides a page like this, which gives a very quick information about the discovery and the background, and it also gives the information about the variant and its associated phenotype, what is the allele name. All those corresponding information about the genetic variant can also be found. And like I had mentioned before, there's an option to download this information in the form of a PDF file. So this is the very first database, the reference and the pioneering database, which is used as a reference for all the blood group related variant information. Other such related databases include the human rhesus base and the blood antigens database. So human rhesus base database is something where we tend to see exclusive details of variants belonging to the RH blood group system. In the recent days, few additions have been made, like variants belonging to the Bombay blood group and the Colton blood group systems are also included. But it was exclusively designed to give more information about the Rh blood group related variants. And similarly, when we look at the blood antigens webpage, it provides a tabulation of all the genetic variants belonging to I think 33 blood group systems, because not all databases are being constantly updated. But this is one of the recent databases which also provides information on the human platelet antigens. In addition to the RBC antigens, it provides information on the human platelet antigens also. And finally, talking about this particular database, BGBAR, which stands for Blood Group Associated Genomic Variant Resource. So this database is something which we will be using in our tasks and exercises also today. So this database is kind of a compilation or collation of all the information which is available in multiple different places. So all of that is collated in this one page, and so that it makes it easier for the user to collect all the information at one particular platform rather than going and searching into multiple places. So before we move on with taking the tasks and exercises, here's a quick link for each of the resources which I had mentioned now. And before going on to the task, let me show a very quick demo of how are we going to surf through each of these pages. Like whatever I explained as a theory now, let me take a very quick demo. So this is the very first database which I was mentioning. That is the ISBT page, the pioneering organizations which maintains the blood variant information. So like I had mentioned, this is the home page. There are multiple tabs which talks about the different components or the information available. We need to go into the working parties tab. So once we do that, it lists all the working parties which are there in ISBT. There's a working party exclusive for the blood components, blood supply management, cellular therapy, so and so. So there are multiple parties, and we are interested in the red cell immunogenetics and blood group terminology. So once we enter that, there's a very quick about the working party section, and we tend to see these three information, like I had shown in the slide also. So when we go into the table of blood group systems, so the tabulation is provided in the form of a PDF. This can be surfed through. This is we can simply surf through this particular PDF page also, we get to see. And there's also an option to download this particular PDF for future reference. Similarly, this page for the antigens and different types of antigens in the system, the tabulation is provided, and there's an option to download the information also. And finally, coming to the blood group allele tables section. So this is how all the blood group systems are listed in this particular page, and whatever we are interested in learning. Like for example, if I want to know about the variants which are present in the Dombrog blood group system, I just click onto that particular tab. So it opens me into a new page. So the first page is all about the history of discovery or whatever, a very quick brief overview of that particular blood group system. And in the next sections, we tend to see we tend to see the list of variants. If I quickly zoom in through. So these are all the list of variants. So like we had discussed in our previous lectures, so a variant can be represented in different formats. So this is one of the formats. So representing the variant in the form of a cDNA position, the nucleotide change, or in the protein position, the amino acid change. So what for this particular variant, what is the phenotype? What is the allele name? Which paper had reported it first? What is the accession number? What is the variation ID? If there's any variation ID, all of this information will be provided in the form of a table. The total, all the variants which are associated with this particular blood group system will be listed here, and the references will also be provided. So if we need to refer this for our future use, this could be downloaded. A download option is also provided here. So this is all about the ISBT data resource. Next, coming to the rhesus base, this is how the home page looks like I had shown in the slide also. So we need to check, we can go into each section, we can check the variants based on their haplotypes also. So this again provides the information in the form of a tabulation. Now coming to the blood antigens page. So this is how the variant information is listed. Like for example, if I want to know more about the Cromer database, Cromer blood group system, and if I want to know about the gene, so I just press into that. So it gives the complete list of all the antigens which have been reported in this particular blood group system, what is the allele frequency in the global population, and all the information which is pertaining to that particular blood group system can be found here. We can also access the sequence of the particular antigen. We can also perform a sequence comparison, etc. And finally, coming and talking about the database which we will be using for our tasks and exercises today. So that is the BGWAR database. So in the right-hand section, there are some quick statistics. When was it last updated? How many antigen information are there and everything. And there's a very quick about the database section. This is the search query where we can search for the desired information. And shown in the example search, we can search for the gene name, allele name, and the variant. The variant can be represented in the form of a nucleotide change or a protein change. Usually these two formats we will be finding in the ISBT resource also, and the DBSNP ID, which is the variation ID. So in this way, we can search for one particular variant if we are desired to search one, or there is a complete section called as the blood groups list. In this has the complete list of all the blood group systems. So if we want to know a complete brief overview of these blood group systems, like for example, I can just press the ABO blood group system. It gives me a complete information, the overall information which we get from multiple sites and multiple resources. Everything is clubbed together so that we get every information at one particular platform. It talks about the history of discovery. Is it a sugar antigen or is it a protein antigen? What is amino acid sequence? What are all the disease associations? How many antigens are present? What are all the names of the antigens? What is the LRG ID of the gene? So what all information, possible information which we will be able to obtain from multiple sources are collated together in this particular site. So once we had a clear idea about what the blood group system is all about, we can view more detail. What are all the genetic variants and their associated phenotypes? So if I want to know about this particular variant, which is associated with a O phenotype, all I need to do is to click on the genomic summary. So I'll be taken into this page, which provides information in multiple sections. So the first section is all about the information about the particular variant. It tells me the gene name, the protein ID, gene ID, what all information I need to know about that particular variant. It gives me the global allele frequencies, what is the clinical significance, is it reported in any particular ancestry, was it a validated information or is it a computational report? All those information will be provided in the upcoming sections. So this is how we are going to surf through each of these repositories, which are very much useful in getting details about the genetic variants which are associated with the blood phenotype. So now we will be quickly moving on to the tasks which we are going to perform in this particular lecture. So now coming back to the exercises, the very first exercise is a very simple task where a variant will be provided either in a nucleotide change format or in a protein change format, and the following information corresponding to that particular variant must be fetched. So the clue here is to use the BGWAR database, use the particular variant which is given in the question, and fetch all the information. So what are all the information that is needed? Gene name, blood group system name, blood phenotype, genomic position, variant type, is it a SNV insertion or a deletion, and variation ID. So the variant which I am showing as a demo is this particular variant, 125G2A. So what are we going to do? All we need to do is three basic steps. We are going to go into the BGWAR website. We are going to paste this particular variant. And once we click on the search button, it will take us to the results page. And from the results page, we are going to fetch all the information which is needed. So all the information is provided in the very first section itself. Like it is shown in the red color arrow here. We can get the gene name, and we can get what is the phenotype which is associated, what is the variant type, is it a SNV insertion or deletion, what is the variation ID, and the genomic position. So here, these are all so when we do that exercise, the information or the solution is the gene name is ACKR1, the blood group system is Duffy, the blood phenotype is FYB phenotype. The gene maps back to chromosome 1 at this particular genomic position. it is a single nucleotide variation, and this is the variation ID of that particular variant. So this is a very quick and simple exercise. And if I would show a demo of this particular example of homepage of the BGVAR website. So we are going to paste the variant. Since I know the variant, I'm just typing in the variant, and we are going to press this search icon. Once we do that, all the information regarding the particular variant pops up. So we get the gene name directly, we get the phenotype directly, we get what type of variant is it, and the genomic position is provided in the nucleotide section. This is the genomic position and the variation ID. And to know more about this particular gene ACKR1, all we need to do is to press this particular tab. So it takes us to the HGNC website and gives more information about this particular gene. So this gene belongs to the Duffy blood group system, and it's in it maps back to chromosome 1. So all this information can be quickly taken from this particular website itself. So based on this demo, the task here is to repeat the same exercise for this variant. The only difference here is the variant is given in the form of a protein change. So the solution for all the tasks which we discussed in this particular lecture is provided so that it's easy for everyone to refer. So the next task here is we will be provided with a list of variants, out of which we need to separate out only those variants that are associated with a blood group phenotype. So the major hint here is that a variant can be deemed as a blood group related variant only if it is spanning in the 51 genes which are associated with the human blood group. So that's the only hint. Consider only those variants which are spanning the 51 blood related genes, blood group related genes. So for the demo section, here I have a list of variants. All I need to do is to copy these variants and we are going to use a new web page here. This is called as the Ensemble VEP web page. So we are going to paste these variants in that particular web page, which I will be quickly showing in the demo also. And we are going to choose certain parameters and download the output file through which we will be able to quickly sort out those variants which are associated with the blood group genes. So this is how the Ensemble VEP page looks like. So it is a very quick online annotation tool which can be used to annotate the variant. Annotation basically means fetching in more information about the particular variant. Which gene does it belong to? Which chromosomal position does it belong to? There are various parameters which can be used to annotate a particular genetic variant. So this is where we are going to paste the input data. So the input data is provided in the form of variant identifiers. So any form of input data can be provided. We can provide a VCF format, we can provide variant identifiers, HGVS notations. So if we want to know what each format looks like, we can just click on it to know how each format looks like. So we have the input format in the form of variant identifiers. So I'm quickly pasting all the input data which we have for our demo, and we are keeping very minimal parameters. Let us choose RefSeq transcripts, and in the identifier we just need the gene symbol. So we will keep very minimal parameters. Find co-known variants, we don't need to know that. Any additional annotations? For this particular exercise, we will not be needing any additional annotations. So I'm just disabling everything. And then I'm just pressing just if you want to check any more examples, and then we can press the run. So this particular exercise will take a few minutes. So here we can see the status of the job. So this job is currently queued, which means it is running. So within a few minutes, once this job has finished running, we'll be seeing the status update. So from queued, it will move to done. So once the job is done running, we will be able to view the output. So in the output, we will be having for each of the variant, we will be able to see the annotations also. Which gene does it belong to? Is it lying in the exonic region or the intronic region? All those information will be provided. So we will also be getting updates on what is the status of the job. So previously it was queued and now the job is running. So now the job is done. So the status is updated here. The job is finished and we can view the results. So all we need to do is just to press this so that we can view the result. So for whatever variant we had uploaded, this is how the results look like. So from here, we can download this particular output in the form of VCF, VEP, or a text file. So I would prefer downloading this as a text file so that we can easily open this in the form of a Excel, and then we can filter out the needed information. So, like I had mentioned, the output can be downloaded in any of the formats. And once we download this as a text format, this can easily be viewed as in the Excel format. And in Excel, we know that we have options to filter. So there is a column called as gene, and in that we can filter only those genes which are coming under the category of blood group genes. So the hint here is we need to consider only those 51 genes which are termed as blood group genes. So these 51 gene list, we will be able to get that gene list from multiple resources. Like I had previously mentioned, the ISBT resource gives a complete list of all these 51 genes. So we will filter out only those genes which are belonging to this blood group gene category, so that we will be left out with only those variants which are present in these genes. So when we do that, out of the 10 variants which I had previously shown, we have only 5 variants which can be categorized as blood group related variants. So this is a quick demo of this particular exercise, and here there's a task. So again, the task is there are 10 variants which are provided. The same exercise needs to be repeated for these 10 variants. So the quick links for this is the Ensemble VEP link, which is provided here, where we have to copy and paste these variants and then just run that particular annotation. And another help or hint for this particular exercise can be found at this particular GitHub page. So in this page, there are options to, there's a particular folder called as the resource materials, where complete list of all the 51 genes and whatever PDF materials which can be downloaded from the ISBT resource are provided in this particular page itself. So anybody who performed this task can go to that particular GitHub page and find references. So now we are done with 2 exercises. We are moving on with the third exercise. So here we have a list of variants which are provided in the VCF format. So we are very well aware by now. We are very well aware of how a VCF format looks like, and out of these variants, we need to predict the phenotype of the KEL blood group system. We know that there are 44 blood group systems. So we are just focusing on one blood group system, which is KEL. So the hint here is that the variant to be analyzed is highlighted in red color. So this is the variant which is belonging to the KEL blood group system. So with the help of all the information which are provided in this particular VCF file, we are going to find out what is the phenotype of this particular individual. So to do that, we need to look into 3 major categories now. So there is a variant. This is the variant which we are focusing on. We need to check the zygosity of the variant. So in the previous example, it is in a heterozygous form. And we also need to know what is the associated phenotype of this particular variant. Since it's an example, I have written the phenotype. It is a KEL phenotype. So phenotype can be fetched from multiple places. So either the person can go and check in the ISBT resource, or they can go to the BGVAR resource, paste the variant, and then they can get the phenotype also. So that is what we performed in our previous exercise. Exercise 1 was all about fetching the phenotype information of a variant. So the interpretation here, the first level of interpretation here is, since the variant is present in a heterozygous form, we clearly know that the person carries 1 copy of the reference allele and 1 copy of the alternate allele. This means they have 1 reference antigen and 1 alternate antigen, presence of both the antigens. So we need to find out the proposed genotype and the proposed phenotype. So the proposed genotype would be 1 copy of reference allele and 1 copy of the alternate allele. This reference allele and alternate allele information can be taken from the BGVAR website itself. So the proposed phenotype would be the presence of both the antigens, the small k as well as the big K antigen. This is how, based on which variant we are analyzing, the genotype and the phenotype can be easily predicted. So with this, so now that we had a very quick demo, this is a quick task. So again, here is a list of variants which is provided in the VCF format. Now, the variant to be analyzed is also highlighted here in red. So for ease or to make this exercise a little more easier, the variant ID is also provided. So all we need to do is to copy this variant ID and paste in BGVAR so that we get the phenotype information of this particular variant. All we need to check is the zygosity. We make our own interpretation and then we are good to go to find the proposed genotype as well as the phenotype of this particular variant. And similarly, it's a very similar task. Till now, we were looking into just one variant. This task is to look into two variants. So like I had mentioned before, all the solution of all these tasks will be provided in the GitHub page. And all the course materials will also be provided in the same GitHub page. This is the GitHub page, the link for which it was provided in the slide itself. So this GitHub page provides all the course materials, like what are all the lectures which we had till date. All the slides will be provided in the form of a PDF. Also, there's a section where we have the solution for each of the tasks which were given. And there is a reference data resources folder where all the PDF files which can be accessed from the ISBT resource can be found here. All the 51 blood group genes related information can be found here, so that it provides a bit of ease when we perform the tasks. We believe this course was useful in understanding the impact of genomics and how are we using the genomic data to computationally predict as well as accurately predict the blood phenotype and the genotype of an individual on an extensive scale. So if there are any queries, we are glad to take and answer those queries, and all of you are welcome to write to this particular email ID which is shown here. And thank you for attending this online course. Thank you once again.